Hello everyone, welcome to sqadia.com. I'm your presenter Sanal Rahman, and today the topic of my presentation is limbs. This is the fourth subsection of this presentation, which is the molecular regulation. It has been further subdivided into the genes which are involved in molecular regulation, the limb axis and the limb bones. Now let's take a look at various genes that are involved in limb regulation. So Hox genes regulate the positioning of limbs along the craniocaudal axis in the flank region. So Hox genes, not an acronym name of a gene, which is responsible for regulating the position of the limbs in the craniocaudal axis. So what is the craniocaudal axis? Cranium is towards the head, caudal is away from the head. So craniocaudal axis tends to be the longitudinal axis along which the limbs have been placed. And Hox genes are responsible for positioning of limbs in the flank region. Flank, obviously everybody knows, it's a layman term, the flank region. So specification of forelimb is regulated by transcription factor TBX5. It specifies the forelimb. Specification of hind limb is regulated by transcription factor TBX4. So what portion of the limb would form a hind limb and what portion of a limb will form forelimb is specified by transcription factor TBX4 and 5. TBX4 forms the hind limb, TBX5 forms the forelimb. So once positioning along the craniocaudal axis has been established, which was done by Hox genes, growth must be regulated along the proximo-distal, anterior-posterior and the dorso-ventral axis. So now we have three different axes. We have the proximo-distal. So if you look at my limb, this is the proximal end, this is the distal end. So this is the proximo-distal axis. Anterior-posterior axis, anterior and posterior, all of you must be knowing what anterior and posterior, anterior forward and posterior backwards. And then we have dorsal and ventral axis as well. For the sake of explanation only, this is the ventral axis and this is the dorsal axis. So these are the three axes along which the growth of the limb must be regulated once Hox gene has positioned the limb bud carefully within the craniocaudal axis. Now, limb outgrowth is mediated by fibroblast growth factor 10. Fibroblast growth factor help in development of a structure, so they help in limb outgrowth. Bone morphogenic proteins induce the formation of apical ectodermal ridge through homobox MSX2. Homobox MSX2 is a gene through which BMP induces the formation of apical ectodermal ridge. Now, apical ectodermal ridge now expresses fibroblast growth factors 4 and fibroblast growth factors 5. These maintain the undifferentiated zone. Now the undifferentiated zone, this is the undifferentiated zone. This is the apical ectodermal ridge which was forming the fibroblast growth factors 4 and 5. Under the influence of this fibroblast growth factor 4 and 5, this zone remains undifferentiated and hence new cells are continuously being formed. Patterning of the anterior-posterior axis is maintained by the zone of polarizing activity. Now this is the zone of polarizing activity in purple. Now it is responsible for patterning of the antero-posterior axis. So cluster of the mesenchymal cells at the posterior border of the limb near the apical ectodermal ridge. As I told you before, these are a cluster of mesenchymal cells which are located at the posterior border of the limb bud and near the apical ectodermal ridge. These cells are called the zone of polarizing activity. They produce sonic hedgehog gene which contributes to specification of the anteroposterior axis. Sonic hedgehog gene, a very important gene, plays a lot of vital roles in the human body development. It also helps in the specification of anteroposterior axis. With limb growth, this zone of polarizing activity moves distalward to remain in proximity of the apical ectodermal ridge. So obviously, the limb is growing, it is moving away from the central axis of the body, that is, the limb is moving distally. Now, the zone of polarizing activity must also move distally to remain in the proximity of apical ectodermal ridge. Now, the dorsal ventral axis. It is regulated by bone morphogenic proteins in the ventral ectoderm. So, the growth along the dorsoventral axis, the dorsal, again for the sake of reference, this is the ventral side, this is the dorsal side. In embryo, the dorsal side is towards the vertebral column and the ventral side is towards the anterior abdominal wall. The growth of a limb along the dorsoventral axis is regulated by bone morphogenic proteins in ventral ectoderm. It induces the expression of transcription factor EN1, again not an acronym. It's just a transcription factor EN1. EN1 expresses WNT7A, restricting it to dorsal limb ectoderm. So the transcription factor EN1, it represses WNT7A, which restricts it to the dorsal limb ectoderm. Now, WNTA induces the expression of LMX1 in dorsal mesenchyme. Again, I know it's a mouthful and a bit difficult to remember, but these are not acronyms and the names of genes which have to be remembered as such. So WNT7A, it induces the expression of LMX1 in dorsal mesenchyme. LMX1 specifies cells to be dorsal. WNT7A also stimulates sonic hedgehog gene expression in the zone of polarizing activity. So it also acts on the zone of polarizing activity and helps in the production of sonic hedgehog gene. Indirectly, it also affects the anteroposterior axis. So indirectly, the dorsoventral axis also affects the anteroposterior axis. So very important are the bone morphogenic proteins in the ventral ectoderm, because they induce the expression of EN1. EN1 represses WNT7A. Repression of WNT7A restricts it to the dorsal limb ectoderm. How does it do that? It induces the expression of LMX1 in dorsal mesenchyme. The LMX1 specifies cells to be dorsal. WNT7A also maintains sonic hedgehog gene expression in the zone of polarizing activity. Hence, it indirectly affects the anteroposterior axis as well. So, the limb bones. So what genes are acting on the limb bones? We've seen this earlier as well. Hox gene determine the type and the shape of the limb bones. Hox expression depends on the combination of expressions of fibroblast growth factors, the sonic hedgehog gene, and WNT7A. With the help of these and with the help of a combination of these expressions, Hox genes are able to determine the type and shape of bone that is required to be synthesized in a certain area. So Hox A and Hox D clusters are primary determinants for patterning of limb bones. So Hox A and Hox D clusters are the primary clusters which are responsible for bone patterning in humans. So in this slide we can also see three types of bones being formed. The limb expression can be divided into three regions. So the first region is the proximal region. In the proximal region we have the stylopods. The stylopods include the humerus in upper limb and femur in the lower limb. In the middle region, we have the zeugopods. The zeugopods have radius and ulna in the upper limb and tibia and fibula in the lower limb. In orthopods, which are the distal regions, we have hand and foot bones. So this was all about this section. In this section, we talked about how various genes help interact with each other and form the adult bone, the adult limb, its bones and its musculature. Hawk's genes were very important. The sonic hedgehog genes were very important. There was a zone of polarizing activity and an apical ectodermal ridge. The limb bud grew in 3 different axes: the anteroposterior axis, the dorsoventral axis, the medial axis. All of these axes are as important as the development of limb bud is. Any defects in any of these axes, any of these genes may result in faulty development of a limb. For clinical correlates, keep watching sqadia.com.